Wonderful. Thank you for the introduction, Dr. Hartwell. Can everyone see my slides? Can everybody see me? Can everybody hear me?
Yeah.
Is that loud enough?
Well, it can be a little.
Is that a room issue or should I shout?
No, it's your name issue.
Can use my outside voice.
Yeah, I
Give me
think
the give me the thumbs up, Dr. Hartwell. Okay, cool. If at any point you need to stop me, ask me to talk louder, by all means, please do so. Um, so hi, I'm Kosta. I've loosely entitled today's talk for the Center of the CSA, PET for Biomarker Imaging of Neurobiological Systems. This is meant to be semi-interactive, so if you have any questions and you're on Zoom, you can just use the raise hand feature or unmute yourself, let me know. If you're in the room, I can't see you, so maybe get Emily's attention. Emily, flag me down. We could stop. I could go over things.
Will do.
But thank you. I've tried to make this as user-friendly as possible, bearing in mind that chemistry is not everybody's cup of tea. Chemistry for chemistry's sake can be kind of boring. So I've tried to focus more on the use of positron emission tomography rather than nuts and bolts organic chemistry. But I think we could probably just start off with something pretty high level. And in the title, I mentioned biomarkers, and biomarker development is a critical part of the drug discovery process. But what even is a biomarker? Anybody have any thoughts, comments, or concerns? If not, I just assume everybody's just so busy thinking, but the dictionary does have a definition of this. It's not just a word I've made up. It's something that's measurable and is often an indicator of a biological state or condition. And they come in many different flavors. These could be proteins, oligonucleotides, saccharides, etc. There's been a lot of money waged in Silicon Valley in the detection of biomarkers. Think back to the Theranos and Elizabeth Holmes story. Um, there's a lot of interest in measuring these things and what they mean for human health. They come in a couple, oops, go the wrong way. They come in a couple different sizes that are familiar to a lot of people. Diabetes does run in my family, so I'm no stranger to this. This would be a glucose monitoring device in which you can measure one's blood sugar level, and that could be an indication of whether or not is somebody has developed or is in danger of developing diabetes or hypoglycemia. But not every pathological state can be measured with something as simple and as portable as a blood glucose meter. And so we look to some slightly more complicated imaging techniques to do that. My favorite technique for at least the past 5 years since I joined Bob's group has been positron emission tomography, and it's used to measure biomarkers in vitro and in vivo in real time and in living systems. Broadly speaking, measuring biomarkers allows for or can allow for the adequate diagnosis of pathology. The white whale, as it were, that PET radiochemists are working on now is using PET to image neurodegeneration disease. And I've incorporated one of these stories into today's talk. I'll talk a little bit about addiction towards the end. The one thing I haven't done is been able to include references. So if there's anything that piques your interest, just let me know and I could, I'll be happy to provide those references. But adequately measuring biomarkers can also be used to predict patients or to select patients that would be predicted to respond to a particular clinical trial or a medication in a clinical trial. And it can also be used to monitor treatment response. And so, again, high level, you can use biomarker detection or measuring biomarkers to accomplish all of this, and this could be all accomplished through PET or a couple different other imaging techniques, MRI, CAT scans, et cetera. The FDA increasingly requires biomarker data to be compiled along with other information in drug development and IND packages. And so again, just a high level overview of how one might develop a drug is there is, of course, an abundance of basic research that goes into identifying the molecular pathways leading to disease. So this could be how the brain changes throughout the process of addiction. This could be how cells undergo rapid division that would be indicative of cancer. That's where I've spent most of my research there and shortly thereafter. Where as a medicinal chemist, I'm interested in mechanism of action. So the pharmacological changes that underpin pathology. And ultimately, the goal of translational research that I find myself in is to compile this evidence, identify a proper drug target, and then use medchem to identify primarily small molecules that could either be used to image that target throughout the course of a disease and or monitor treatment response. And if you're lucky, you can, get something into clinical trials. Through Bob's U19, I've got an interesting ligand that might go first in humans, so I'm pretty excited for that. And that's looking at alpha synucleopathies and Parkinson's and other neurodegenerative diseases. Um, but moving from preclinical to clinical translation is quite a feat that can take many years, millions of dollars, et cetera. But the role that PET plays in that is to enable, again, that adequate biomarker measurement. And so how PET works, without getting too into the weeds, so just as a high level overview, PET, again, is this technique in which we'll often combine a small molecule, and these small molecules are what I designed. So these small molecules will often include a fluorine 18 or carbon 11 or other different synthetic radioisotopes that emit positrons. And so that's indicated. Let me pull up my laser pointer here. That's a pen. That's not a laser pointer. Indicated by this beta plus. So this beta plus is a positron. And when a positron collides with an electron, that results in a process known as annihilation, and a pair of photons are released, such that if this small molecule that is radiolabeled is in your body and circulating and accumulating in a target of interest, the ensuing annihilation and photons that are emitted can be imaged and captured in a PET scanner. And so here we have a cute little picture of, perhaps that's me with the shaven head, and envision that I have had a PET tracer injected into me that accumulates in my brain. That PET tracer then emits photons, which are calculated ideally on our pen PET Explorer, and then through complicated reconstruction processes, you get in vivo real-time data of where that radio tracer is accumulating in vivo. And that can be indicative of various different pathological states, or it can be used for monitoring treatment response. This is what a PET scanner would look like. If you've ever had an MRI, and I've had quite a few, um, it is a little claustrophobic to envision yourself going in there, but these come in a couple different flavors, couple different sizes. But again, this does enable the biodistribution measurement of your PET radio tracer. And for my research, again, I'm primarily an organic and medicinal chemist. I'll use medchem to develop a small molecule that will bind selectively to a protein or target of interest. Then I'll go back, radio label them, and then evaluate these radio tracers in different kinds of models, often starting with non-human primates, primarily to evaluate things like brain penetration. Getting a radio tracer into the central nervous system is quite challenging. You've got all these efflux transporters that do a good job of keeping exogenous chemical matter out, which must be overcome in order to develop a proper PET radio tracer. So I thought I'd just start with, I have three stories. I could talk for a long time about all this, but just three stories that might be of interest. Not really focusing on the weeds of how we do radio labeling. I welcome anyone, should you want to come to the cyclotron, we could do that. We could talk chemistry, so on and so forth. But really, I just wanted to give you a flavor and a taste of all of the things that PET can do. And as I alluded to, the white whale, or one of the biggest competitions in PET radiochemistry right now is the evaluation of amyloid targeting therapeutics and enabling the early detection of various different neurodegenerative diseases. So neurodegeneration being a blanket term incorporating Alzheimer's, Parkinson's. These are often caused by the misfolding of one or many different kinds of proteins. And being able to identify this protein misfolding at an early stage is key to allowing a physician or a clinician to intervene therapeutically, where you might have the best, best progression-free survival, or the best clinical outcome. And this has been, as you can imagine, a very expensive endeavor, taken not just by the pharmaceutical, but also the academic scientists. But there is a big payoff. Various different foundations, such as the Michael J. Fox Foundation, is promising a $1 million cash prize to the first team that develops a PET radio tracer that can detect alpha-synuclein. And this is a misfolded protein that is implicated in Parkinson's. To date, there is one approval for treating AD that is caused by amyloid beta or the misfolding of amyloid beta protein. And so just as a high level overview of the pharmacology, amyloid precursor protein is something that you and I have inside of us, but through various different semi-natural processes and or through exposure to oxidative stress or reactive oxygen species. This amyloid precursor protein can be cleaved and summarily can aggregate, forming these oligomers followed by plaques. And this, again, high level, is thought to be the cause of cognitive decline. And so much research has focused on developing plaque targeting, amyloid beta plaque targeting, either small molecules or antibodies, with the goal of reducing these plaques in vivo. And this has been great. However, if you've been following the clinical literature carefully, even though it would appear that aducanumab or Aduhelm can clear these plaques, there's really no cognitive benefit. So there is no restoration of cognitive ability. And the role that PET has played in this has been pretty astonishing. There are a couple of different PET radio tracers one would use to image amyloid beta. And this goes back to the mid 2000s. So, for context, I was in middle school when a lot of this work was done. But this is carbon 11 PIB, Pittsburgh Compound B, developed by Klunk and Mathis at Pitt. And this is a carbon 11 radio tracer that binds selectively to misfolded amyloid beta. And so you can use this not only to triage patients to identify which stage of the disease they are on or in, you can also use this to monitor treatment response. Whereas if you have a patient at baseline that shows uptake of the radio tracer, following treatment shows diminished uptake of the radio tracer, you might say that they've responded favorably to that treatment. These radio tracers do come in a couple of different shapes and sizes. So PIB is a carbon 11 labeled radio tracer. Carbon 11 is a short-lived radioisotope. Half-life is only about 20 minutes, which really precludes PIB from being transported pretty far from a cyclotron facility. Whereas fluorobetapir, which is labeled with F18, has a much longer half-life, and this can be transported much further from a cyclotron facility where you could have your PET scanner in slightly more hard-to-reach spaces. So summarily, the goal is to identify radio tracers that are compatible with these longer half-life F18 radio labels. I'm going to move my camera box here. Uh, And here is just an image indicating, or rather demonstrating, just the utility of these PET scans. So this is a 79-year-old patient that was imaged with fluorobetapir, and these intense colors simply just show radio tracer uptake, indicating a high likelihood of misfolded amyloid beta protein. And this is usually cross-validated with immunohistochemistry. And in fact, the only way to confidently diagnose many of these cognitive decline diseases is post-mortem, right? So we take brain sections and we perform immunohistochemistry to evaluate what proteins are present in brain section. And this patient did indeed have a high beta amyloid burden, which indicates further high likelihood of amyloid beta protein and Alzheimer's disease. In using PET, again, we could triage patients and or diagnose them earlier in the disease process where therapeutic intervention might still, provide better quality of life and a longer life. And these are some seminal studies that have appeared in the literature where you can look at these are antibody amyloid beta targeting antibody treatments where we have a treatment group at screening and at week 78. And so these are amyloid beta targeting PET tracers that are used. And you can sort of see on the left with the initial screening, we have high uptake sort of in the front of the brain here. And then following a couple weeks of treatment, you see less uptake of the radio tracer, indicating treatment response. A little bit more pronounced in the second pair of images here, where we can see almost complete clearance of these amyloid beta plaques, which is reflected by the lack of radio tracer uptake. And if we compare that to a placebo, we can see that it actually looks like as the disease progresses with placebo treatment, you have an increase of this radio tracer uptake, which is likely caused by progression of the disease. So these proteins spread almost through a prion-like process where misfolded proteins cause more proteins to misfold. And this has been reproduced and seen in the literature with various different amyloid beta radio tracers and with a couple of different amyloid beta targeting small molecules and antibodies. But again, this is just a high-level overview of how you can use PET to monitor treatment response, particularly in neurodegenerative diseases, which again is a pretty hot area with many different proteins drawing many different groups' interest: amyloid beta, alpha-synuclein, TDP-43, et cetera, all causing a variety of different proteinopathies. Any questions on neurodegeneration in PET? I have two more stories I'd like to share. And again, happy to go as slow or as fast as people like. But if no questions, I'm happy to keep going.
Yes, there's a question.
We have a question. Cool.
You said that you could only diagnose, like confidently diagnose with, I think Alzheimer's it was in that example, post-mortem with other imaging. What else could be a cause for the misfolded proteins in the scans while the person was still alive?
That's a great question. And this is another hot area of research. So we know misfolded proteins, either amyloid beta, alpha-synuclein, generally cause and or are antecedent markers to developing cognitive decline, which and these can, I should mention, these can be diagnosed via many different behavioral tests and cognitive tests. Uh, but we're still kind of learning what causes these proteins to misfold in the first place. And there is a emergent theme in neuroimaging, and that is the need to image neuroinflammation and reactive oxygen species. So there are PET tracers that can do this, but it's not exactly clear yet what proteins are implicated in that. For example, there's a protein translocator protein that has drawn a lot of interest, and there are a couple PET tracers that can be used to image that. That's associated with neuroinflammation. But then there's also PET tracers that can image reactive oxygen species. So this happened a long time before I joined Bob's group, but Bob has a PET tracer called Ross Trace that can be used to image oxidative stress that is generally associated with aging, but this can be caused through other exposure to different environmental factors. And it's also thought that TBI, traumatic brain injury, can cause, excuse me, various different proteinopathies and or just other cognitive deficits somewhere down the line. Does that kind of answer your question? I'm not seeing a nod, but I'll take that as a yes. Okay, great, great. Don't let the glasses fool you. I'm still kind of blind, and people are sitting far away. Any other questions on neurodegeneration?
I have a question.
Yeah.
I thought that the whole like beta amyloid plaque hypothesis around AD was like somewhat discredited. So can you talk about that or speak to that?
Yeah, I can tell you it's a hot area. It's a hot area of research, and there are different proteins that misfold and can give you different proteinopathies that lead to um, different kinds of diseases. So, amyloid beta being one protein, detecting it via with PET is useful, but it was found, and in lending to the discredit of that particular theory, is that clearance of these plaques did not really restore cognitive benefit. Um, However, that's not to say that they're still not useful. And some people think that being able to identify the disease with PET earlier in the disease or process or in the prodromal phase might still have better outcomes. But people are, in addition, looking at various different other proteins. What's one particular confusing factor about these proteinopathies is that you can have one protein misfolding in many different ways, which will change radio tracer uptake. And further, these will present with different clinical symptoms. So if I had it my way, being able to design one PET tracer for each protein in its each misfolded form would be illuminating, and I think could provide a lot of information. But that is going to be a multi-year, probably multi-lifetime project. You could argue we're kind of there with PIB and florbetapir, and a couple of other proteins too, but I think it still leaves much to be desired. So I do agree. Amyloid beta is, I think, a contributing factor, but I don't think it's the only contributing factor. And I think other proteins do play a role, particularly with neuroinflammation and reactive oxygen species.
Well, thank you.
Sure. Oh, I have a
There
chat.
is a question in the chat.
Are you planning to look at tau as well? Excellent question. Yes. Yeah. So tau, misfolded tau, either 3' or 4' tau, that is a protein that can be misfolded and is implicated in, again, various different proteinopathies, Parkinson's, also Alzheimer's, honestly. And so another hot area of research, one I'm interested in, is designing these small molecules that bind specifically to this protein. As you can imagine, the work is quite expansive. You really need a team to do it. Also helps if you have a lot of funding. Not a great time to be a scientist looking for funding from the NIH, but uh, yeah, I think this does draw a lot of private foundation grants too, particularly the Michael J. Fox Foundation, looking at tau and alpha-synuclein um, as being just a whole slew of proteins that you could really look at to triage, identify, and diagnose different cognitive diseases. I forgot to mention tau. It was a good catch. Anything else on neurodegeneration? I'm happy to come back. Should people, if I say something, you're like, I had a question about that. Else, I will go on. So this was a high level using PET to monitor treatment response and or diagnose a patient with a neurodegenerative disease. Another use of PET that I find quite interesting is using PET to calculate receptor occupancy. So in drug design, a medicinal chemist must ask a couple questions. Is the drug metabolized? Where in the body does the drug go? How do we know it gets there? And it turns out you could use PET to answer a lot of these questions. So one series of studies that I think illuminates this point quite well is the development of a migraine medication. And the protein of interest is calcitonin gene-related peptide receptor, or CGRPR. Love a good acronym. And here we're going to use PET to evaluate whether or not a small molecule engages that protein. And so just a high-level overview of migraines. I suffer from migraines. And one of the pathways that lead or is thought to lead to migraines or contribute to the pathophysiology of migraines is that this protein is endogenous, calcitonin gene-related peptide receptor. When this protein is activated through calcitonin gene-related peptide, it triggers a migraine. And so this is a G-coupled protein receptor, which is activated through a peptide, giving you a migraine. Lots of acronyms. But if we can develop a small molecule that can preclude this protein's activation, that might be a useful treatment for migraine. And indeed, several FDA-approved medications for migraine target this protein. And so the questions that, this was a study performed by Merck, the questions that Merck asked were, can we use PET to determine where this protein is located in vivo? Turns out it's expressed in the brain. And can we evaluate the effective dose needed to effectuate migraine relief? So how the study was designed is the following. We're going to take a carbon-11 PET radio tracer, which binds our protein. And so here we have carbon-11 MK4243. This can be administered and as total body biodistribution measured using PET. This is going to elucidate the biodistribution of calcitonin gene-related peptide receptor protein. And the two small molecule antagonists, or the two small molecule putative medications that were evaluated, were talsegapant and MK3207. And so what you could use, you could administer this PET tracer and sometime later administer one of these small molecule antagonists, which were developed as anti-migraine medication. And you can evaluate each molecule's ability to kick off or displace the PET radio tracer in vivo. And this gives us an idea of the receptor occupancy of each small molecule. With the idea being each medication has what's known as a therapeutic window. So you need to administer a certain amount of a drug to elicit a therapeutic response. If you administer too much of the drug, you get off-target binding and side effects. So there is sort of this magic window with which you can administer a drug where it's thought to be ideal. And you can use PET to measure that. Also, in addition with PET, we often use PET's cousin known as autoradiography, and this makes use of a longer lived half-life. And so instead of a carbon-11 radio-labeled isotope or radio-labeled small molecule, you can use a tritium-labeled molecule. So this uses tritium-labeled MK4232, sorry. And these are human brain and rhesus macaque brain tissue where we can evaluate radio tracer binding. And we can see that this radio tracer binds to certain areas of the brain, such as the substantia nigra, the brainstem, cerebellum. And this has both, this is cross-validated again in rhesus monkeys and humans. And this is often done to confirm where the PET signal in vivo is originating from. And so this is, again, just indicative of where a PET radio tracer would bind. And this matches the distribution measured through Western blot analysis. So similarly, how we'll use immunohistochemistry to measure the presence or absence of amyloid beta protein, you can use Western blot to measure protein expression in various different areas of the brain, which can then be cross-validated with autoradiography in vitro and PET in vivo. So these are all kind of similar techniques we deploy in the drug development process. Now, what Merck did was to take various different concentrations of the drug, administer them to either monkeys or humans, and measure their drug plasma concentration. So this is as simple as just inserting a catheter, pulling a plasma sample, centrifuging it down, and calculating, usually through mass spec, how much drug is in the blood. You can then correlate that with receptor occupancy as measured through a PET scan. And so this is a baseline carbon-11 PET radio tracer scan. And again, this is a calcitonin protein targeting radio tracer. And we see high uptake, particularly sort of in the back, the occipital region of the brain here. But when we administer a large dose of a calcitonin protein receptor antagonist, we see that the radio tracer uptake is diminished. And you can do some mathematical modeling and calculate what dose is needed to sort of kick off or preclude the binding of this radio tracer, such that you can get these really elegant relationships between plasma concentration and protein occupancy, such that you can elucidate something known as the therapeutic window. Here are some additional images, and these are clinical images taken in human human brain using PET. And so here's a baseline scan using our carbon-11 radio tracer showing high brain uptake. If you administer 140 milligrams of, this is talsegapant, you see there's virtually no difference between baseline and 140 milligrams. And I'd like to note this is an efficacious dose. This is enough to alleviate migraine symptoms. So what we can therefore conclude is that you could administer a relatively small dose of this drug to alleviate migraine, such that you only occupy about 5 to 10% of the receptor. And again, less is generally more with the drug. The less you could administer, the less likely you are to cause off-target binding and further different side effects such as nausea, et cetera. And this is particularly advantageous in clinical oncology, where the administration of lots of drug can lead to things like hair loss, nausea, et cetera. You can see it takes almost tenfold excess of an efficacious dose to really displace the PET radio tracer, where your receptor occupancy is between 40 and 60%. And so a lot, busy slide, but the take-home message here is, again, just that we can use PET to illuminate and calculate receptor occupancy of drugs in vivo. And again, this is real time, which is pretty unheard of for any other imaging modality. Any questions on the use of PET to illuminate drug binding in vivo? This works both for the central nervous system. So talsegapant is a blood-brain barrier penetrant molecule, as is the PET radio tracer used in this study. But you could also use this with instruments like the Pen PET Explorer, which is a total body PET imaging instrument. There's only about two in the United States, so it's becoming more common to use these kinds of studies in drug development, not just for CNS targeting drugs, but also peripheral nervous system targeting drugs as well. Any questions, comments, concerns, or bitter remarks? If not, I'll continue. And what I have next, I think, is the most interesting topic of the three stories that present today. And so there's been some really elegant studies using PET to evaluate the brain distribution of the 5-HT2A receptor, and also for using it to calculate these receptor occupancy studies for psilocybin. So many of you are probably aware that psilocybin is an efficacious treatment for treating lots of different disorders, and this has only been recently explored in the last decade or two, right? So there was a large pause on using classical psychedelics to treat many diseases from about the 70s to the mid 2000s. But recently, various different groups at Hopkins, NYU, Penn, Yale, University of Toronto, et cetera, are looking at classical psychedelics. So that's psilocybin, that's DMT, that's LSD, for treating not just major depressive disorder, but also substance use disorder and smoking cessation, various different maladies. This is taken from the Compass study, in which it was discovered that treatment with either one or two doses of psilocybin led to a meaningful and statistically significant change in depressive score. And so on the y-axis, we have weeks out from psilocybin administration. And here we have, on the y-axis, we have change in your MADRS score, where the 25 milligram treatment arm shows a statistically significant and sustained response. Again, that lasts upwards of 6 to 8 weeks, which is pretty astounding. And this is often from one or two administrations of the drug. I know a lot of drugs as a medicinal chemist. I don't know many drugs that can do this, cause this lasting and profound decrease in score. If you think about classical antipsychotics, antidepressants, SSRIs, usually these are administered once daily for a very long time. Yes, that is, beneficial to the pharmaceutical company manufacturing them. But if we can get higher throughput to patients that suffer from depression or substance use disorder, that would unobjectively be a good thing, right? Where you can sort of cure a patient of their depression following just one treatment. Also being mindful, this isn't a snake oil that can cure everything, but I think this certainly merits further investigation. What a group in Denmark did, and this is under the stewardship of Gitte Knudsen, a PET radiochemist in Copenhagen, was to use a carbon-11 radio-labeled PET tracer. And so this is SIMBI 36, which was developed to image the 5-HT2A receptor. And she used it to calculate these dose response curves. And so there's a lot going on here. But I'll first just, demonstrate or I'll present to you how the study was designed. And so patients were dosed with psilocybin, either 3, 6, 12, 15, 18, 24, or 30 milligrams at 0 minutes. Following psilocybin administration, patients underwent a carbon-11 SIMBI 36 PET scan. And so here you have a 5-HT2A targeting therapeutic, psilocybin, followed by administration of a 5-HT2A targeting PET tracer, SIMBI 36, at 60 minutes post-dose and 344 minutes post-dose. And what this allows you to do is measure the clearance of the therapeutic in vivo in real time and calculate these receptor occupancy studies. Patients were also administered a subjective effect questionnaire post-study. And so this can take a couple of different a couple different shapes and forms. Often the MEQ, mystical experience questionnaire, is used, where you're asked questions such as, did you feel a sense of oneness one with the universe? Do you feel that you met God, etc.? And so what this allows for is a correlation between the dose administered, the intensity of the feeling. Patients were asked to kind of rate their experience from 0 to 10, 0 being, it's whatever, this was a normal day, 10 being, this was the most intense experience of my life, and so on and so forth. And another component that was tied to that was receptor occupancy of psilocybin. I'd first just like to give sort of a top-down view of the pharmacology of psilocybin. So it should be noted that psilocybin is not a drug. It's actually a prodrug. When magic mushrooms are eaten or otherwise ingested, this phosphate group is cleaved, which results in the formation of psilocin. And this is the active 5-HT2A targeting therapeutic. These can be metabolized in a few different pathways, but suffice it to say, if we can measure the receptor occupancy of 2-psilocin, which is a 5-HT2A targeting agonist, we can start to correlate this with various different outcomes, including, change in depressive score, receptor occupancy, so on and so forth. And for those that are not aware of this and or who haven't tried mushrooms, which is everybody, often, there's a 20 to 60 minute in which it come up, if you will, right? So you ingest the mushrooms between 20 to 60 or 15 to 30 minutes, give or take. You reach a high in which you might start to hallucinate. There's a plateau effect which lasts between 2 and 4 hours. There's a come down after administration. And then the after effects, which can include hallucinations, last between 1 to 24 hours. And this is dependent on the psychedelic. So psychedelics such as DMT are thought to have a shorter dose-dependent after effect, which is advantageous because they could, in theory, be administered with a little more high throughput. Psilocybin, what I've read is it's somewhere between 8 and 24, whereas LSD has a pretty prolonged after effect, lasting upwards of 24 hours. And as an aside, the discovery of LSD is a pretty interesting story where it was accidentally ingested. The chemist who made it accidentally ingested it in the 50s, rode his bicycle home, was hallucinating, thought his neighbor was the devil. He journaled about it extensively, but in the pharmacology community, there's a day, I think in April, dedicated to his accidental discovery of discovery and ingestion of LSD. But again, just goes to show you how potent and unique some of these drugs are. All right, let's get to some of the nitty gritty. And so these are plasma concentration levels measured over a period of time. So various different subjects, again, were administered various different doses between 3 and 30 milligrams. Blood samples are pulled every couple minutes and plasma psilocin concentration measured. This follows a somewhat as expected path, as expected outcome, where generally the more psilocybin that was ingested, the higher the plasma concentration of psilocin was observed. And the inverse is also true. The less psilocin ingested, the less or lower the plasma concentration measured was observed. Interestingly, this also is kind of reflected in the intensity rating. So you have your low dose, 3 milligram administered patient. It looks like sort of, reaching a plateau, an intensity rating of about 4 out of 10, which is still pretty, it's a pretty interesting outcome, right? 3 milligrams is not a lot of drug to be administered, but and still to have, an experience rated as 4 is pretty intense. And what we could see is definitely the more drug that was administered, the more highly patients rated their experiences in terms of outcome. With it looks like, what is this? The 24 milligram, maybe the 18 milligram, all between that 8 to 10 range. Again, bearing in mind, less of a drug usually is better for minimizing off-target binding and side effects. What PET was used to do was to correlate the plasma concentration with the 5-HT2A receptor occupancy. And you can sort of see this as a plateau effect where patients administered, roughly 18 milligrams hit this 70-ish% occupancy, which isn't really that different from patients administered 30 milligrams. So there is definitely an upper threshold to what looks like what can be reached in terms of receptor occupancy. 3 milligrams, still rated at a 4, but didn't obtain all that high receptor occupancy. Um, and of course, receptor occupancy can be correlated again to the intensity rating. So lots of different ways these can be sort of cross-correlated, cross-validated through pulling plasma, measuring drug concentration, using the 5-HT2A PET receptor SYMB36 to measure um, GPCR occupancy, and also correlating that to intensity scale, which is pretty neat. What was found is that it looks like it takes about 1.95 or about 2 micrograms per liter of drug to be administered to reach about 50% occupancy or to get an OC50. And it looks like at about 0.42 mgs per kilogram milligrams per kilogram, it's likely that you achieve greater than 70% of receptor occupancy. Again, which isn't a lot of drug, all things considered. So you could administer 18 milligrams or 30 and still sort of get the same desired clinical outcome. And if anybody's so interested, this research group has done a lot of interesting studies showing that not just that you can measure the receptor occupancy studies, but you could also measure the downstream effects of psychedelic administration. So there's another PET tracer that I won't have time to talk about today because I didn't have time to incorporate the slide. But the long-term effects of psychedelic administration are thought to be mediated by increased synaptogenesis. And a protein implicated in that is the synaptic vesicle glycoprotein 2A. And as you can guess, there is a PET tracer for this protein, and that PET tracer demonstrates increased uptake following psychedelic administration. And so Gita Knudsen in Copenhagen has done this in various different preclinical models. I think using rhesus macaque and pigs, showing that protein remains elevated for at least 7 days after, again, acute administration of psilocybin. Um, which is really paradigm shifting. Not to belabor the point or beat a dead horse, but the fact that you could get lasting, chronic relief from acute administration is disruptive technology, to put it lightly, and is really, illustrates an exciting time in pharmacology research. And this is sort of just taken from the Compass study in discerning or distinguishing and deciding sort of what dose to go with. It was found that the high dose, the 25 milligram, did have a higher proportion of sustained response. So there might be, in addition to reaching, minimum, let's say 70% occupancy, there might be some other factors at play, which, delineate the difference between the fact that more patients have a sustained response the more they take in comparing the 25 milligram treatment arm to the 10 milligram treatment arm. But suffice it to say, it's still really impressive to see that you have many different patients administer different doses of the drug, demonstrating this long-term relief from depression. And this has been extended to various different maladies, substance use disorder. Let's see, Michael Bogenschutz at NYU has a really interesting study with alcohol use disorder. Matt Johnson, formerly of Hopkins, has done some really interesting work with smoking cessation. And I'm really curious to see where sort of this work will go in the next couple weeks, months, years. If you're a nerd like me and peruse clinicaltrials.gov on the regular, if you type in psilocybin, DMT, LSD, there are currently hundreds of studies that are ongoing, and several of which include the use of PET to measure not just receptor occupancy, but how the expression levels of different proteins change as a result of psychedelic administration, and how the expression levels of that protein remain chronically or longitudinally. So that's really that's all I had. I'm happy to engage any thoughtful discussion in the next 15 or 20 minutes. I guess I did have a few more slides here. Yeah, just again, kind of beating the point here, not to beat a dead horse or belabor the point, but there is some, inter individual variability. Not to belabor a point, but I really think PET can be useful in selecting patients that would be predicted to respond favorably to treatment. And I pulled a clinical oncology example out of my hat, but I really think it could also be useful for predicting patient or identifying patients that would respond to psychedelic treatment as well. And with that, I'll yield the floor for questions, comments, and discussion.
Are you going to the psychedelics conference?
Which psychedelics comp is this Philadelic?
No, the next week one in Denver.
Oh, I am not.
Okay.
I'd like to. I'd like to go. I've not been to Colorado.
Yeah, me neither.
You're our ambassador, Claudia.
Yeah, I'm the ambassador. Are you going?
Enjoy.
Yeah, I'm going.
There's also a psychedelic reading group next Wednesday, if anyone would like that information, over at Wharton, I believe, but I can forward that to anyone who's interested in going Wednesday at lunchtime. Other questions?
I have another question. What are you excited about in like pet research and the stuff that you're doing? What do you think is like the next exciting, interesting thing?
So I'm a little biased just because it's my particular project, but I have worked on a pet tracer that can image alpha-synuclein in vivo, and it's got a lot of really good properties. It binds with high affinity. It exhibits really good brain uptake and rapid clearance. And so we are getting that tritium radio labeled in Sweden now. It's going to get shipped back to us. And then we take brain sections from the Penn Biobank, and these are clinical cases. So we're going to evaluate this tritiated radioligand in Parkinson's, Alzheimer's, so on and so forth. We already have a lot of preclinical PET imaging done, where it shows really, exemplary uptake in rhesus macaque models. And should the autoradiography look good, first in human studies would be, as every medicinal chemist dream, to sort of get something in humans. The rule of thumb is it usually takes 5 to 10 years. I've been a postdoc for 5 years, so who knows? Maybe this is my big break. If not, I'll find something else to be excited about. My initial interest was in using PET to image the mu opioid receptor. And it perhaps could be noted that Dr. Hartwell, myself, and Dr. Jay Wright from radiology have a small grant where we're developing a new radio tracer to image the mu opioid receptor, which that radio tracer has advantages over carbon 11 carfentanil. I could talk a lot about that radio tracer and just how crazy it is, um, but I don't want to venture too far off course. But it's something else I'm excited about.
Cool.
Yeah, what are some other imaging methods other than PET that are used in the field?
There's been a lot of done work with MRI and fMRI, and Robert Carhart Harris seems to be the person to do that. And okay, there are a couple other groups, but what they could do is they can evaluate how functional connectivity in the brain changes as a result of psychedelic administration. So I just happen to be a PET guy, and I think PET is great, but I do think it's important to have different information acquired through different imaging modalities to get really a whole picture of how a mechanism of pharmacology is is taking place. CT scanning as well. I don't think X-ray would be all that useful, but in no particular order, PET, CT, and MRI seem to be the most commonly used in the group.
I just want to plug my lab that from UCL. I did my PhD in UCL, and we're doing the biggest fMRI study on DMT at the minute. And I did like part of my PhD on that. So like I just wanted to like shout out to my lab at UCL in London.
Woohoo! When those are published, please disseminate to everybody on the call. I'm really curious as to what
Yeah, I'm
findings will
hoping
be.
to get like loads. It's called the Unity study, so it's understanding neuroplasticity induced by tryptamines. And my supervisor is actually opening a psychedelic research center at UCL. So he has funding for that, so that's going to be like a big thing as well.
Awesome. My fingers are crossed for your advisor's behalf. That sounds really cool. And if they need a pet guy, just maybe connect us or don't.
Yeah, I still speak to him.
Shameless plug for shameless plug.
Any other questions?
I have another silly biology question. So if you're um, targeting receptors in a certain part of the brain, but then those receptors are found everywhere in the brain, how do you like send the drug to just one region? Is
That
there
is a
someone
great
understanding
question.
something wrong there? Like, is this an
G-
actual problem?
Generally speaking, if you have a target expressed ubiquitously in the brain and administer a drug that targets that protein, it will go everywhere in the brain. Radio tracer uptake is governed by, does it bind to the protein or not, and does it cross the blood-brain barrier? But for example, I have a project looking at the delta opioid receptor, which is in different areas of the brain, of course. And I can't yet get that radio tracer to bind selectively in the front of the brain versus the back of the brain. I do think that would be a really interesting project or an interesting thing. But what you can do is target different, they're called isoforms of the protein. So if you have, in the example I'll pull just for simplicity, is neurodegeneration. If you have misfolded amyloid beta or misfolded alpha synuclein in the front of your brain and a PET tracer that binds selectively to the misfolded protein, that's where the PET tracer will go. If you have normal alpha synuclein or amyloid beta in the back of the brain, that tracer will not accumulate. And the way we design a lot of our assays to evaluate this is cleverly, we will take the misfolded protein and clone it and or let it grow, replicate. We'll do our experiments in vitro. These experiments are called radioligand competition binding assays, and we'll evaluate the PET tracer's ability to bind to those misfolded proteins, not necessarily and or not at all the native protein. So when I do preclinical PET scans in just a monkey, the outcome we're looking at isn't does it bind to the protein of interest really. It's does it get into the brain? How quick it does it get into the brain, and how quick does it wash out? Um, so that's actually a very good question. It's something it is kind of on the forefront of PET research. Targeting different isoforms of a protein.